Before molecular biology became an established field, it was universally accepted that simple organisms, like the E. coli bacterium, were incapable of any manner of sexual reproduction, which, for prokaryotic purposes, describes the simple exchange of DNA through some kind of cell-to-cell contact. But if asexual reproduction is the only way for bacteria to reproduce, how do you explain the extraordinary ability of these organisms to adapt and remain resilient despite only being made up of a single cell? Joshua Lederberg's journey into medicine and science began early on in his childhood. Born in 1925, he was a first-generation American child of Palestinian parents and raised in Manhattan by a father who was an Orthodox rabbi and a mother who was descended from a long line of rabbinical scholars. Lederberg developed a passion for science at an early age. At 7 years old, he expressed his desire to become like Einstein. His father, who had originally hoped that Joshua would continue the family tradition of religious service, eventually came to terms with his son's interest in science. By high school, Lederberg was recognized as something of a prodigy, where his interests focused on biology and he conducted his first experiments in cytochemistry. Lederberg later described his early pursuits as being driven by an unswerving interest in science, as the means by which man could strive for an understanding of his origin, setting, and purpose, and for power to forestall his natural fate of hunger, disease, and death. In 1941, Lederberg entered college at Columbia University. Mentored by biochemist and gifted teacher Francis J. Ryan, his passion for biomedical research flourished. In 1943, Lederberg enrolled in the Navy's V-12 program as a pre-medical student, which put him on an accelerated track to become a doctor for the armed forces. He completed his undergraduate curriculum in zoology in 1944 and enrolled in Columbia University's College of Physicians and Surgeons that same year. Amid the scientific fervor surrounding the recent discovery by Oswald Avery that deoxyribonucleic acid, or DNA, is the genetic material, Lederberg spent his first year of medical school continuing to conduct research under Ryan's supervision. Carrying on with his research in early medical school was highly unconventional. However, Lederberg was eager to continue his study of bacterial genetics. He was particularly motivated to prove the classic binary fission model of reproduction is not the only one used by bacteria, and that they are not genetically identical generation after generation. After several failures in his research, Lederberg sought the guidance of Edward L. Tatum at Yale University. Tatum was a previous advisor of Ryan's and an expert in bacteriology and the genetics of microorganisms. Lederberg took a one-year hiatus from medical school in 1946 to carry out experiments with the intestinal bacterium E. coli. This is when he made the discovery that bacteria do indeed exchange DNA. In the experiment, 2 different mutated strains of E. coli were used. Each strain was incapable of synthesizing an essential amino acid in minimal media and would not grow individually on its respective plate. When combined with the other mutated strain, however, the media produced progeny and successfully colonized. This discovery would alter what scientists had always believed about bacterial evolution and the acquisition of new properties, such as antibiotic resistance. It also proved that subsequent generations of bacteria are genetically distinct. Lederberg extended his leave from med school and collaborated with Tatum for another year to map the E. coli chromosome. He submitted his research on genetic recombination in bacteria as his doctoral thesis and received his PhD degree from Yale in 1947. In 1958, at just 33 years old, Joshua Lederberg was awarded the Nobel Prize in Physiology or Medicine, along with Edward Tatum and George Beadle. Following his receipt of the Nobel Prize, Joshua Lederberg would go down in history as having co-discovered not one, but 2 of the 3 mechanisms by which bacteria transfer DNA. In the 1950s, he collaborated with Norton Zinder to discover a method where DNA was exchanged without the ability of the bacteria in the experiment to conjugate. This was the mechanism of transduction. Joshua Lederberg was not only a pioneer of molecular biology, but an extraordinary scientist across several fields, from his groundbreaking research on E. coli in the 1940s and 50s to his technological and biological advances in astronomy, medicine, computer science, and even public policy.
The effective scientist, the really creative one, is the person who 1 minute can be fantasizing with no restraints, imagining all kinds of things, blue sky, and the next can be his or her own harshest self-critic.